Okay. Now we will move to the IMViC tests. So I think you all have come across IMViC tests and you have done it in the laboratory or at least read it. So here it consists of a series of 4 tests, which are like which the name suggests, as in I for Indole, M for Methyl red, V for Voges-Proskauer test, and C for Citrate test. So why is this IMViC test done? This is done to differentiate between different Enterobacteriaceae or coliform bacteria. Okay, so what are these bacteria? These are present in our gut or in our surroundings, like in soil or in water. So you will find gram-negative bacteria all around yourself. So these are the Enterobacteriaceae family. These belong to those family, and these tests are helpful in differentiating those. So a coliform, as you have see, as it is written, a coliform is a gram-negative aerobic or facultative anaerobic rod which produces gas from lactose within 48 hours. So this is a definition of the coliform bacteria. So these IMViC tests are designed in such a way so that it is able, like you will be able to differentiate between different Enterobacteriaceae and coliform tests. So the first one is Indole test. In this, we check if the bacteria is able to produce the enzyme tryptophanase. So what does this tryptophanase enzyme do? Please pay attention, this is slightly complicated. So the tryptophanase enzyme here will break down the amino acid tryptophan. So how will it do that? There are like 2 steps of breaking down tryptophan. So the first step is the formation of indole pyruvic acid from tryptophan, and then the tryptophanase enzyme catalyzes the deamination reaction, and it just shifts this amine group, and this is the pyruvate that separates, and we get 3 different products, which are indole, pyruvate, and ammonia from tryptophan using the enzyme tryptophanase. Okay, and pyridoxal phosphate is required as a coenzyme for this reaction. So now, like as you know that you have to add Covax reagent. Like first you have to inoculate your tryptophan as tryptophan broth, and then you have to incubate them. I mean incubate the uh, tests, and then you have to add 5 to 6 drops of Covax reagent. So here the Covax reagent has a compound called dimethyl aminobenzaldehyde. Okay, as you can see in this reaction, in the second reaction. So here, in the first reaction, the bacteria has tryptophanase enzyme and the indole is produced. In the second, when you are adding this reagent, I mean the Covax reagent in your test tube, then indole and this p-dimethyl aminobenzaldehyde will react with each other in the presence of HCl and butanol, and then the red violet compound, which we can see as a cherry red ring in the top of the test tube, that you will get. So can someone tell me, like, why do we get a cherry ring and not the entire media gets colored like red colored when we put Covax reagent in the media? If you have read it somewhere or if you know.
Guys, do you know the answer? Or if you don't know the answer, you can say we don't know. We're not going to judge you.
Like, we are here to help you, so don't worry. We kept this session as a fun session, so you don't get bored, because we know that understanding these reactions and all can be tedious for you. So okay,
Okay.
like I'll tell.
Don't know, please explain.
Okay, sure, I will. So what happens is, when you add Covax reagent, it only doesn't have p-dimethyl aminobenzaldehyde, but it also has HCl and amyl alcohol. Okay, so amyl alcohol, as you know, is insoluble in water. So that's the reason why you get a cherry red ring, because it's insoluble in the media. So like it's only present in the, it will be floating in the like water or in your media. So you will be getting a cherry red ring, and not the entire media will get colored. Okay, so I hope that this is clear now. So let's move to the second test, which is the methyl red test here. So in this is a pretty simple test. Here, as you know, that glucose is converted to pyruvic acid through glycolysis. Then pyruvic acid can be metabolized into several acids, using the mixed acid pathways. There are so many mixed acid pathways through which it can produce acetic acid, lactic acid, succinic acid, formic acid, etc. So when these acids are formed in the media, the pH of the media is drastically reduced. As you know that acids are capable of reducing the pH or decreasing the pH. So the pH will go from neutral to 4.5 or around or below, like, which is acidic. So when you add methyl red, which is yellow in color, it will turn this yellow color into red color. Okay, because of the acidic environment of the media, it will turn itself red in color. So the positive test is red and the negative test is the yellow. Like your media will stay yellow even after the addition of methyl red. So this is the MRVP broth preparation. This is for 1 liter of your media. So like the MRVP, both methyl red and the Voges-Proskauer test both have the same broth composition. So there's no different media composition for both of this. You have to stick to this buffered peptone glucose and dipotassium phosphate, and you have to prepare the control tubes and your test tubes, and you have to incubate them. So as you can see in this picture, as I explained, that glucose is first converted into pyruvic acid, and then the pyruvic acid goes into several mixed acid pathways and produces lactic acid, acetic acid, etc., which reduces the pH of the media, and that's the reason why methyl red changes its color from yellow to red. Here, E. coli will give you a bright red positive coloration, and Klebsiella, on the other hand, will give you yellow coloration, which is negative in this case. Now we come to the third test of the IMViC series, which is the Voges-Proskauer test. In this, we test if the organism is able to produce acetone or not. Okay, the other name for acetone is acetyl methyl carbinol. So after glucose fermentation, if the bacteria is able to produce acetone or not, we will be checking that. Okay, so first I'll explain you what happens in the reaction. So first glucose is converted into pyruvate, as you know, like via glycolysis it metabolizes to pyruvic acid. Then via butanediol pathway it produces acetone. So this butanediol pathway now has two enzymes. The first is acetolactate synthase, which uses two molecules of pyruvate and converts it into acetolactate. And then this acetolactate is converted using the enzyme acetolactate decarboxylase to acetone. Okay. So first, two molecules of pyruvate will be used by acetolactate synthase to produce acetolactate. Then acetolactate will be converted to acetone using acetolactate decarboxylase. So here you are checking if your microorganism has these enzymes and is able to convert pyruvic acid to acetone. Now, first I'll explain you the method, then I will go to the second reactions and everything. So here the method, you have to inoculate the MRVP broth as I explained it, and then you have to incubate it. After incubation for 24 to 48 hours, you have to first put 6 drops of 5% alpha naphthol, which is also known as the Barrett's reagent A, and then you have to keep it for 5 minutes. Then you have to add 2 to 3 drops of 40% potassium hydroxide, which is Barrett's reagent B, and then you have to shake it and keep it for 10 minutes, and then you have to observe. Here also you will get a pink or a red color when it's positive and yellow color if it's VP negative. So why does this color change take place? I will explain that to you now. When after acetone is formed and you are putting both the reagents A and B, Barrett's reagent A and B, the Barrett's reagent A, what it does, it acts as a color intensifier. Okay, it acts as a color intensifier and it catalyzes the reaction. But the second Barrett's reagent, which is Barrett's reagent B, which contains KOH, okay, potassium hydroxide, it converts acetone to diacetyl, and then diacetyl will react with the guanidine component of peptone. Like you are adding peptone in the media which has guanidine component, it will react with that and it will form the pink red color in the presence of alpha naphthol. So that's the explanation why you have to add Barrett's reagent A and B to get the results. Here, as I told you, that you will get a red color if it's positive and a yellow color if it's negative. I hope that's clear. If you have any doubt, you don't please don't hesitate to ask. And okay, this is the last test for the IMViC series and the last test for today's session, which is citrate utilization test. In this test, you will be checking the like you will be giving bacteria the sole source of carbon will be citrate and the sole source of nitrogen will be ammonium salts. Okay, so what happens in this reaction is if the bacteria has citrate permease, it will be able to use this citrate and it will convert it into pyruvic acid, and then the bacteria can freely metabolize pyruvic acid into whatever it wants. Like in the other scenarios, we are adding glucose or several other carbohydrate sources through which it is converting that into pyruvic acid, and then again through pyruvic acid it is going on to further metabolic reactions. But in this case, citrate permease is required because you are not adding any other carbohydrate source unless, except for citrate. So it must have citrate permease so that it can use it. And if it doesn't have citrate permease, it won't be able to grow in the media. Okay, you won't get any growth itself. Okay, so now what happens is when the bacteria is using these ammonium salts, okay, it is breaking down these ammonium salts and it is converting those ammonium salts into ammonia. So as you know that ammonia is responsible for increasing the uh, pH, or like it increases the alkalinity of the media. Hence the bromothiamol blue, which is the indicator over here in this test, like in the media bromothiamol blue is added. So bromothiamol blue will change its color from green to blue because the pH has changed from neutral to basic. Okay, so you have to make sure that you remember that which tests have acidic pH, which tests have basic pH. So this is one test where you have basic pH, and because of that the color is changing from green to blue. Okay, so this is the media composition for citrate test. Generally, we use a ready-made media which is known as Simon citrate agar, which comes as a ready-made box for citrate test. But if you don't have access to Simon citrate agar, or if you don't have it in the laboratory, you can use uh, these components and you can prepare it in the laboratory. It's like the same thing basically. So here, as I told you, that citric acid will be converted into, using citrate permease, it will be converted into oxaloacetic acid first, and then pyruvic acid, and then it will metabolize into several other reactions. So the method for this is very simple. You have to prepare a slant, okay, Simon citrate agar slant, and preparation of slants we have explained it to you earlier. So you will just prepare a slant and you will streak it from the end till the mouth of the test tube, and then you will incubate it at 37 degrees for 48 to 96 hours, because citrate is one test where like getting results takes time. You take you like it will take time for bacteria to metabolize and give you result, like change its color from green to blue. But you also have to make sure one thing, that your controlled test tube, okay, that should be green in color even after 96 hours, even after like 4 or 5 days, whatever incubation period you are keeping for. Your test might change but your control shouldn't change. If your control changes, that's when you understand that this test can't be processed anymore and you have to discard everything. So here, as you know, the results is blue for, I mean, green to blue, and the negative result is the original deep green forest color. Okay, so these are the IMViC test results. As I have explained it to you, the Indole test has cherry red ring, and the negative one has yellow ring, or you might not get any ring. Or the MR test has red color, the MR negative has yellow color, the VP has again red color, negative has yellow. Citrate has negative has green color and positive has blue color, changes from green to blue. This is a IMViC test chart for different microorganisms. You when you are performing it in laboratory, this chart might be helpful for you. You can see like E. coli is positive for the first two and negative for the last two. Staphylococcus first and last negative. and the methyl red and Voges-Proskauer test is positive for Staphylococcus aureus. So likewise, it's given for all the organisms, and it's not universal, but almost. I mean, for 99% of the cases, when it's methyl red positive, the bacteria is generally Voges-Proskauer negative. Like, exception is Staphylococcus aureus because it has both positive MR and VP positive. But other than that, like even if you go for other microorganisms, if one tests positive, methyl red positive, the other test, which is VP, might be negative. It's not universal, but yeah, it's like that. So that marks the end to our session today, and I hope that you have enjoyed and understood all the tests we have explained it to you. Please, if you have any doubts, you can ask us. Please don't hesitate. If am I audible?
Yeah, now you're
Yeah,
audible.
network. So, like, yeah, if you have any doubts, you can feel free to ask us, or you can just type it in the chat box. If you want us to explain a topic once again, we can do that. And if you have any doubts, you can ask us in the WhatsApp group as well. I hope that you have joined it. And for the previous sessions, you can watch our YouTube videos.
So you guys have no doubts?
Okay,
Either we were too good with the presentation, or you didn't listen to what we said. Those are two extreme possibilities. And we are being positive. We were good with our presentation. So,
Okay, can you tell me, like, which one did you find the most interesting? If someone can tell me which test was the most
which test do
interesting
you
one?
think is most interesting? You can type it out, we'll read it.
Yeah. Okay, we explained catalyst, recapitulate. We explained catalase, oxidase, coagulase, um, motility
What
test,
did you say?
motility agar, and use of seam media for like three different tests. H2S production, of course, IMViC tests, which includes Indole, Methyl red, Voges-Proskauer, and Citrate test. So, which one was the most interesting?
Have you guys performed this during your college
Laboratory day,
time?
motility
Okay,
test.
motility test. Have you got the results when you have performed it? Because when we were, I'll tell you this personal thing that when we were doing motility test, not once did we get the results proper. Either we
Yeah,
add too much of our media onto the cover slip, or we are observing it under the microscope because of the cavity slide, we break the cover slip.
yeah,
So
yeah.
all these things have happened. So according to me, motility is the toughest of all the tests.
True, true, true. While performing, it's the toughest
Yeah.
because the like media should literally hang in the cavity. So that
Yeah,
is the
it
most
should hang. Otherwise, you won't see the organisms moving.
yeah.
Only one response.
In,
IMViC test.
okay.
Nice.
Okay, so hope you have understood everything. And tomorrow again we will meet at 5 o'clock, and we will explain you all the other biochemical tests
Remaining
along with
biochemical
yeah
tests.
along with API system and whole genome sequencing as well. So please do join tomorrow. We hope to see you all again.
And you can give your valuable feedback to us on the WhatsApp group because we are still learning with you guys. So your feedback is going to help us a lot.
Yeah.
Um, that's all. The link to the WhatsApp group is in the chat box right now. You can join. If you have
Okay.
not joined the group, please join and stay connected with us. Okay, guys,
Okay.
I think we'll end the session here.
Yeah. Thank you so much, guys, for joining. Hope to see
Thank
you
you,
all tomorrow.
guys.
ಹೆಂಗ್ ಬರೋದಪ್ಪಿ